Hi, everybody. My name is Mike Reapy, and I am the president of the UC Santa Cruz Alumni Association. I'd like to welcome you tonight to our monthly speaker series, Slugs and Steins, Lectures from UC Santa Cruz. For those of you who are new, our Slugs and Steins series engages a UC Santa Cruz faculty member in discussions with you, the local community of Silicon Valley, and our extended community online, with the goal of making all of us Renaissance women and men. We wanted to feel just like you're at UC Santa Cruz sitting in class, but with drinks. So David, another volunteer organizer, he's with me tonight. We're both alumni and spend our days in entrepreneurial companies, Silicon Valley style. We'd like to thank our usual host, San Jose's Forager Tasting Room and Eatery, for 2 years of support. Of course, they're closed down right now because of COVID, and we're joining you online. But we really hope that we'll be together with Forager and our friends there soon. Tonight, we raise a virtual Stein with research scientist Dr. Peter Weiss-Penzias, who will lead us through his fascinating research about mercury, mountain lions, and our own coastal food impacts. Peter received his PhD from the University of Washington in chemistry in 1995 and has worked as an associate researcher in the Department of Microbiology and Environmental Toxicology at UC Santa Cruz since 2009. He is an internationally known researcher of mercury biogeochemistry with over 2,500 citations, according to his Google Scholar profile. We'll take your questions after the talk, perhaps a few while we're talking, in Zoom's Q&A box. So you can add your questions there. You'll be able to see everyone else's questions. You can upvote them if it's similar to yours. You can also add comments and we'll incorporate that. So you don't need to wait until we ask. You can submit your questions at any time and that'll help us stage them, and we'll do our best to answer them in the order that they come in. This talk is being recorded. In a few days, you'll be able to find it on our UC Santa Cruz Arts and Lectures YouTube channel. We'll post the link in our social media and in the follow-up email that you get if you registered online. So does everybody have their Stein? I've got your slug, Dr. Peter Weiss-Penzias.
All right, thank you, Mike. It's a real pleasure to be here. Thanks for inviting me, and too bad I did not get a chance to do this at the actual Forager, but we'll make the best of it. So hopefully I can give you some new information, make some connections that you hadn't thought about before. The topic here is mercury in the environment, which is a toxin and something that we should all be concerned about. And we're going to talk about it in a rather unusual place, which is fog. I've kind of cornered the market on mercury in fog. If you Google the words mercury and fog, you will come up with a lot of the reports of my research. Not too many people into this kind of thing at the moment, but it is a growing awareness among atmospheric and biogeoscience researchers. Where is mercury? It's turning up in some unusual places. So I'll go ahead and get started here and give you an outline. I'm going to start with some basics, some background on mercury. Mercury is an element. It's part of the earth. It is natural, but humans have disturbed it, have liberated it, have emitted it, and that has caused all sorts of other processes to be accelerated since the Industrial Revolution mainly. And so we'll talk about emissions, deposition, methylation, and bioaccumulation. Oh, some long words. And so then I'll introduce mercury and fog, which will connect the coastal ocean to the terrestrial biosphere. And then specifically, we'll look at mercury in a coastal terrestrial food web. And that's like a food chain where the big thing eats the little thing, which ate the littler thing. And we'll look how mercury moves its way through the food web and try to answer the question, do we see evidence of mercury delivered through fog? And then I'll talk about my latest research, which is has this catchy title, There's Mercury in the Fog, so what about our food? We'll look at the potential risks to people. As I'll explain, we realized during this research that no one has ever really looked at the mercury content of foods grown in foggy areas. So if there's mercury in fog, how much could be getting in our food? Okay, so let's start off with the first unit here. And hope everyone's comfortable, got your stein in your hand. And so let's talk about some biogeochemistry. So we are thinking about mercury in the atmosphere. So mercury is everywhere, right? It's part of the earth, it's in soil, it's in our bodies, it's in water, but it's also in the atmosphere. It's the most unusual metal. It's liquid at room temperature, and liquids evaporate. And not only that, when mercury is subject to high temperature, it goes directly into the air and it remains there as a gas. A single atom of mercury is in the gaseous form. And so the red arrows going up are what we call emissions. So they're putting mercury into the atmosphere. That includes biomass burning. Vegetation takes up mercury from the atmosphere. So when it's burned, it liberates it. And geogenic emissions, those look like the Egyptian pyramids, but they're, I guess, supposed to be mountains. So volcanoes, mercury-enriched rock and soil will release it back to the atmosphere. And then we've got coal combustion, metal mining, smelting, and cement. Those are the big 3 or 4 different types of mercury sources that humans are causing. And then there's ocean emissions. Now, interesting thing about what's coming out of the ocean is a lot of that was previously deposited, previously fell out of the atmosphere from human emissions. So that's not entirely natural. Okay, so we'll put some numbers on these. And you can see that the coal combustion, metal mining, smelting, and the ocean emissions are the largest. Then, of course, we have this process called deposition, which is taking it out of the atmosphere, and I'll talk more about that in a moment. But let me just draw your attention to what we'll also talk about more in a moment here, which is the methylation by anaerobic bacteria. And that's what we're really interested in for the health effects, because mercury, when it's in the elemental mercury form, it's not nearly as toxic compared to when it's in its methylmercury form, and that is accomplished by anaerobic bacteria. Okay, some fun facts about elemental mercury, gaseous elemental mercury in the atmosphere. The total amount in the atmosphere is about 6 billion grams. Of course, the atmosphere is very large, so that works out to a concentration of about 1.5 nanograms per cubic meter of air. And that's on the order of parts per quadrillion. So it's extremely dilute. And each mercury atom stays around in the atmosphere for about 1 year. Okay, now what happens to it after in this year time period? Well, it gets removed by oxidation and wet deposition. So straight up chemical reaction in the atmosphere, and there's many possible oxidants of elemental mercury, but the main one, it seems at this point, is this bromine radical, which is just a bromine atom. It has an unpaired electron, and it finds a mercury atom that's in its elemental form, and it oxidizes it into what we call the 2 form, the mercury 2. It's been oxidized. It lost a couple of electrons. That mercury 2 is then very reactive and sticky. It can be taken up by cloud droplets. So it finds some water in the atmosphere, and then it quickly dissolves, and those cloud droplets grow, and they fall back to the earth as rain, snow, fog, and also as dry particles. But those are harder to measure. Okay, so we have some data on this rain out of mercury to the surface of the earth, thanks to the National Atmospheric Deposition Program, Mercury Deposition Network. And this has been a network of sites across the US and Canada, and now it's international. Started in the late 90s. And so that every year they generate a map of deposition. The units here are in micrograms per square meter. So it's just amount of mercury falling as rain or in rain per square meter of land area. And color-coded, more reddish gives you more and green is less. And the pattern here is somewhat reflective of just the amount of rainfall. So that's why you see a lot of mercury depositing up here, because it just rains a lot there. And but in the Midwest here in the South, it does rain a lot, but it still has also very high concentrations of mercury in that rainfall, which is not entirely understood. The pattern here is quite a bit different than what we'd see for our typical air pollutant and something that gathers in rainfall, which is the sulfate ion, and this is essentially acid rain. And we see a hot spot in acid rain kind of right here where you would expect, because it comes from sulfur dioxide, which is from burning of coal. And you've got, Indiana, Ohio, Pennsylvania, and the most sulfate there. But you don't really see that for mercury. You can see it's not necessarily very low, but it's not the highest that it is compared to like Florida, for example, where there's very little coal-fired power plants. So the difference here is that mercury is much longer lived in the atmosphere, and what you're seeing is atmospheric processes that are scrubbing it out from higher in the atmosphere for mercury, and that represents more of the global background. The sulfate ion is pretty much coming from local emissions or regional emissions, whereas mercury is coming from much further afield, what's all mixed together in the global atmosphere. Okay, so that makes improving or lowering mercury deposition a bit more challenging, because it requires some international cooperation. It doesn't mean that the US emissions aren't important, but they only add to the global pool of about maybe 5 to 10%. Okay, well, we're really concerned about health effects here. This process of making methylmercury is one of nature's great accidents, I think. It doesn't really serve any purpose. Perhaps for the bacteria that do it, they may be trying to detoxify, but no one's actually quite certain why bacteria do this, but they do it, and we suffer the consequences. So mercury gets into mainly aquatic systems, and it can come in mostly from the atmosphere, but some can come in through runoff and rivers, but the atmosphere is more important for delivering mercury to water. And the mercury mixes around and it enters into a bacteria in an anaerobic or anoxic environment, and methylmercury is produced. Now the mercury coming in to the bacteria is very low concentration, and the methylmercury coming out is also very low concentration. However, methylmercury behaves more like an organic pollutant because of that, just the chemistry of it. It sticks to fat cells. And so what happens is you get something called bioaccumulation. Okay, you've got dissolved in water, maybe parts per quadrillion level, I mean, very dilute, and then goes into some phytoplankton, which would be parts per trillion, some zooplankton, which would be parts per billion, and finally up to fish, which can be at parts per million level. And of course, human exposure at this concentration is just unhealthy. And we'll talk about why that is in a moment. But it's kind of amazing that we get this 1 billion times enhancement just by a natural process. So, mercury in the environment, in water and in air, is extremely dilute, parts per quadrillion. I mean, you can't even hardly fathom that, it's so dilute. But yet just this kind of accidental process enhances it up to levels that are no longer, that we can just say, no big deal, because it is a big deal. Okay, now there's been a lot of research on methylmercury in fish and aquatic environments. It's very, very heavily studied. Much less research on this other product of methylation. You can see slightly different chemical compound right there, and that's called dimethylmercury. So the bacteria, they'll make monomethylmercury and they'll make dimethylmercury, and dimethylmercury just usually reverts back into monomethyl. But in the coastal environment, especially where we have upwelling, and I'll talk more about that in a moment, some of this dimethyl can just go out into the atmosphere, okay? And so Normally that wouldn't be a big deal. Would just kind of react back to less harmful forms and fall back into the ocean. But where we have fog, it appears that the dimethyl gets absorbed by the fog. Now, just a word about dimethylmercury. It is so incredibly toxic. A researcher died working with dimethylmercury, and we don't even work with it in the lab. I've never even seen it. It's just not something you even want to get your hands on. It's volatile and it's extremely toxic. So it's very difficult to contain. And but it makes researching it quite interesting because, you know, I mean, if you're going to do research on toxins, right, you might as well pick a good one. And dimethylmercury is certainly a good one. Okay, so just give you a little bit more detail here. And I can take a break after a few slides to answer any questions on this. I know this is a lot of like chemistry and biology, and all this mercury intersects many different disciplines. And that's why a lot of us mercury researchers really find it fascinating. So this is another area of active research. How do microbes methylate mercury? This is just one cartoon that illustrates one process. There are some other ones, but it appears that in something like a biofilm substrate, so this can happen within the water column or in sediments, an anaerobic environment is created, so lack of oxygen, and thiols are produced from sulfate. There's a lot of sulfate naturally in water, and in an anaerobic environment, you're going to have thiols, which are a reduced form of sulfate. And that can complex with mercury to make this mercury thiol complex, and that apparently makes it easy for it to get into the cell. And once it's in the cell of a anaerobic bacteria or an archaea, the genes are listed here that code for these proteins. The gene is called HGCA, and that puts a methyl group onto a mercury, and then the HGCA protein gets sort of regenerated by the HGCB. And so more details are emerging about this process. But what we really want to keep our eye on is its health effects on animals and humans. So we are kind of like top predators. And so we eat animals that have eaten other animals. And that can expose us to a lot of methylmercury bioaccumulation. So the part of the world that is experiencing the most risk to this right now is in the Arctic, where you have native peoples whose diet relies upon animals that are high on the food chain. So beluga whales, polar bears, seals. And so these are some mercury concentrations in the brain. And this is in micrograms per gram and wet weight. So it means that just as sampled, they didn't dry it out. And this scale here is logarithmic. And so the amount of mercury at this line right here is the observed effects on the NMDA receptor. Basically just means when they cut open the brain, they saw some changes. So when the mercury level in the brain was up to 0.1 ppm, parts per million, that's when they started to see some neurochemical changes. And then the neurochemical threshold where they would see some toxicity effects are right around 1 part per million in the brain, okay? And so you can see that for some animals like beluga whales, many of the samples exceeded that threshold. For humans, not all of them exceeded, but the error bar indicates that some of them did exceed that threshold, meaning that there was imminent danger. And how this affects people, certainly it affects the person who has ingested too much mercury by damaging their brain. But the biggest risk is for the developing fetus. So in the brain of the developing fetus, we have the neurons, and at the end of the neuron is the growth cone as those neurons are developing. And the mercury interrupts the growth cone and makes it inactive, okay? And so if too many of those growth cones are inactive, then the neuron doesn't work and the baby is born with severe birth defects, neurological problems, and that is called Minamata's disease, named after a great industrial accident in Minamata Bay, Japan. Well, actually not so great, really infamous is the proper word. Okay, and so just to stress, mercury has no known biological benefit. It just messes with cells. It binds to sulfur very, very strongly, and sulfur is one of the key elements in 2 of the amino acids, cysteine and methionine, and those amino acids are essential for some of the 3-dimensional structure of proteins, and so mercury can really interrupt the 3-dimensional structure of proteins. Okay, so I noticed a couple of Q&A questions, so that's kind of the background to mercury. I can go ahead and take a couple questions at this point and then introduce my specific research.
Thank you, Peter. So we have a question from Tiffany. How old were each of the species sampled? I'm assuming that the humans were old.
Yeah, I'd have to go look at the paper to relay that information accurately. What we do know that mercury accumulates with age. So older people and older animals tend to have more mercury. We tend to ingest it faster than we can get rid of it. It does leave our bodies in our hair. It binds to the protein in our hair, and we're constantly sloughing off mercury. But if you eat a lot of fish and seafood, marine mammals, not many of us do, but certainly the Arctic natives, you're going to be ingesting a lot more over time.
Good, thank you. We had one shoot to the top here. Tom asks, are you aware of any evidence of methylmercury getting into groundwater, perhaps through anaerobic bacteria in porous substrates?
Yes, that is an area of research. So I'm not aware of all the examples, but I know in coastal lagoons, like our Younger Lagoon at Long Marine Lab, the lagoon is a kind of an incubator for anaerobic bacteria, and the mercury gets methylated there, and then it can percolate as groundwater and enter the coastal ocean that way. So that is thought to be one of the contributors to methylmercury in the water column of the coastal ocean. Thanks.
And Peter, we have a question from Daniel Fernandez.
Yeah.
Sounds like he's a chemist. Hi, Peter. In the ocean, you seem to show Hg0 and Hg2. Are both of them what are eaten by the bacteria? And are there other exchanges between mercury 0 and mercury 2? In other words, does mercury 2 revert and become mercury 0? And when does this happen? Also, is there an important distinction between mercury 0 and mercury 2 in terms of its chemical reactivity or danger to living things?
Yeah, so that, again, a whole other area of research. Mercury undergoes very complex changes in seawater, in all water, between mercury 0 and mercury 2. So mercury 0 is essentially our safe mercury. It's not as bioavailable, and it also is not very water soluble. So it's going to leave the ocean pretty quickly. If it gets into the mercury 2 form, then it can complex with those thiols and enter into the bacteria and become methylated. So mercury 2 is really the key. It can fall out of the atmosphere directly in rainwater, and it can also be formed within the ocean. Some of it can revert back to mercury 0, but that process can be kind of slow. Okay.
One last question from Meg. Is the red line of the neurochemical threshold for all of the different types of brain cells or only one animal type?
I think those apply to mammals in general. Now, of course, every mammal is going to have some differences. So these are just kind of gross estimates. There's probably more information available for health effects on humans based on some accidental poisonings that have happened through the years. And so I could find some more specific numbers for you if you wanted to contact me after the show. Okay, great questions. All right, so mercury and fog, this is where it gets a little more specialized. So, but the interesting thing is that the ocean is affecting the land. And we know that it does. I mean, obviously it affects the climate. If you're, in Santa Cruz or San Francisco Peninsula and you see these giant redwood trees, this is a function of our oceanic climate. But fewer people realize that the ocean actually can transfer nutrients and toxins, if there happen to be toxins in the ocean. That coastal slice of land that is most exposed to the ocean can receive elements and compounds that are emitted in sea spray or gaseous compounds that come from the ocean. And even more so if there's fog, then this can be another vehicle to transmit oceanic emissions to the land. Okay, so I first thought about this after reading a paper that came out of UC Santa Cruz lab, the one that I ended up working in. So as they mentioned, I started around 2009, and that's when this paper came out. Chris Conway was a graduate student of Russ Flegel here in the UC Santa Cruz. He's now retired. Russ is, and I've taken over his lab space and still working on mercury with some of the same instruments that Russ first procured. And this paper mentioned dimethylmercury. They were out on a ship and they measured dimethylmercury concentrations in seawater as a function of depth. So depth in meters, you can see this is at the surface, and the depth at 200 meters. And they were at the 3 different locations. So C1, C2, or M1 and M2, those are buoys out in the bay. And so then the concentrations of dimethylmercury are increasing that way. And you can see that at the surface, there were some, oops, sorry. Let me go back. There were some enhancements where it was greater than 0 at all 3 locations. And if you look at the time of year of those cruises, it was the open circles, and those are March and May, springtime. Well, what happens in the springtime is something called upwelling. Okay, the winds start to really kick up. In the atmosphere out over the Pacific, we have this high pressure system. It rotates clockwise, and that essentially pushes the winds in that direction. They go from north to south, and that's what pushes the water and gives us the California current, which is a cold current because it's basically coming from Alaska. It's coming straight down the coastline. And so that's why it can be, hot in Santa Cruz, and you go swimming in the ocean and the ocean's really, really cold because that water was way up north not too long ago. But there's another phenomenon that's occurring as well, and that's called upwelling, which is also bringing cold water to the surface. Okay, and that works because of the Coriolis force. The Earth is spinning, but it's not spinning as fast at the poles. It's spinning faster at the equator. Okay, if you've been on a merry-go-round, if you're at the center, it's not going as fast. If you're out on the edge, it's going much faster. And so the water that is going from north to south actually gets deflected in that direction. Okay, it goes like that. It goes offshore, right? And so that creates a sucking of the water offshore. So there's like less water right near the coast. And if you look in the vertical direction, that's going to cause upwelling to replace that water, right? The water's getting sucked offshore because of the Coriolis force, and it has to be replaced from something. So the water gets sucked up from below. Well, that's where you have a lot of nutrients. That's a good thing, right? That's what promotes all the rich marine life that we have in Monterey Bay and other places where we have a lot of upwelling. But it also brings methylmercury because methylmercury is produced in anaerobic environments, and those are at depth. Okay, so could this dimethylmercury be evading to the atmosphere as a function of upwelling? That was the idea. Okay, now in addition to upwelling, and the season of upwelling, you also have fog. So I put together, this is, found this little time lapse on the internet, thanks to Emeric, and kind of looped it here. And to explain, okay, you got the high pressure atmospheric conditions, you got subsiding air from aloft, sinking air, and that causes warming. But at the same time, you have cool air because the sea surface temperature is like 55 degrees. And if you were to look at the vertical profile of the atmosphere, you'd see this inversion, okay? And that prevents vertical movement and creates a marine layer at the surface where gaseous water condenses into tiny cloud drops, and hot rising air in our valley creates a pressure gradient and wind, which sucks the coastal fog inland, okay? And so putting together the dimethylmercury upwelling out of the coastal ocean with the fog and having a sort of an intimate moment on my morning ride to UCSC, I literally came up with this idea. I wonder what is in these fog droplets. I think I probably was wiping the water off of my glasses, and it's like 52 degrees out on the 1st of July, and I'm like, this weather kind of sucks. It's supposed to be summer. And I was like, what's in this stuff? And realized once I looked in the literature that no one had ever measured methylmercury in fog before. So this was the hypothesis for the atmospheric chemistry and the process. You got dimethylmercury evading from the coastal ocean as a function of upwelling, dimethylmercury in the atmosphere being a rather unstable compound, encountering fog droplets, which are naturally acidic. The marine aerosol has a pH of about 5, or can be as low as 3 due to ocean emissions of sulfur. And that would make monomethylmercury, which would be a lot more stable in the fog because it's an ion. And then as the fog drifts inland, it would drip that monomethylmercury to the forest and the surface. Okay, and then if you think about how important fog is, I mean, it's a bit ephemeral and ethereal, but in some locations, west-facing ridges like Montara Mountain in San Mateo County, there was an early study by Goodman in 1985. And you can imagine if you've ever been above Montara or like San Bruno and these places where the wind is just whipping that cloud up the mountain, you get this orographic lift, which cools the cloud further, causing basically a drizzle effect. So those fog droplets are coalescing and it's essentially raining. They were able to get 7.1 liters per day of fog water as an average over this time period. 2 different locations, 2 different collectors, 3.5 liters per day, and we're using another collector. And so this is kind of, as an aside, is like, wow, this is maybe an untapped resource for some water. But anyway, that's a lot of water. If there's methylmercury in it, that could really impact the ecosystem. So that was the hypothesis going into this. And the question was essentially, do we see evidence of mercury from fog in this coastal terrestrial food web? And so I was pondering this one day and met up with actually my neighbor, Chris Wilmers, and was talking to him about my research, and it was explaining that, my hypothesis that I wanted to see if there was any evidence of mercury in the food web due to fog deposition. And he said, well, I have a bunch of mountain lion fur and whisker samples that we've archived from our investigations of mountain lions. And would you like some? And I was like, okay. And that's how science works. So Chris's group has done some extensive investigations of mountain lions, the Puma concolor, and this is from a trail cam in the Santa Cruz Mountains. And that looks like, you know, one of the trails right behind UCSC, and certainly there have been mountain lions spotted there. And so they have many samples he was able to share with us. They also track the mountain lions with the radio collars, the GPS signals, and the accelerometers. So they know their position, they know that some of their behaviors. They've tagged many of the mountain lions and given them these names. The M stands for male. They also have a long list of females as well. And so you can see that they are out there. This is 2015, a 5-day period, and that's the ones they had tracked at that time. And one of them, 36M, actually came into Santa Cruz. And so this is a little close-up map here. One of 36M's forays into town, he tended to come every 2 to 3 months at night, spend the day under thick vegetation, and leave the following night. The time between these time points is 5 minutes. And I just thought I'd put up where my house is. You can probably figure that out if you want to come visit me. I'm up there around the edge of town. It's pretty nice place, but I actually did see a mountain lion, and it could have been that one. He was running across the road right here. It was about maybe 5-6 years ago. So that could have been 36M. They're awesome creatures, but you know, you got to think a little too close to home, right? You're looking over your shoulder as you're taking a walk in the early morning. Okay, so and then the fog frequency can be shown by this map right here. So this is summertime fog frequency from satellite images, sort of a composite, and the colors represent the hours per day. So the dark blue is like 14 hours a day. So out here in the ocean, it's like foggy more than half the day on average during the summer. And inland, it's, you know, 0 hours per day. And across the Santa Cruz Mountains, starting at the coast, you can see Greyhound Rock and Younger Lagoon. And we had some other sites as well. Very foggy, upwards of 8 to 10 hours per day on average of fog. And then this Blue Oak Ranch, actually that star is a little bit misplaced. It should be down here, more in the red area. They're getting maybe 2 hours, but if that. Of course, any sort of fog or marine stratus that you would see at Blue Oak Ranch would be very much higher and not precipitating, whereas the fog at Greyhound Rock and Younger Lagoon is oftentimes getting everything wet. So we utilize this information to kind of design our study sites where we were going to collect vegetation to see if there was an influence of mercury in the vegetation from fog, and I'll get to that more in a moment. Okay, so here's the results of the study. We had a bunch of mountain lion whisker and fur samples, and the blue ones came from Chris Wilmer's group, and the black ones here came from the California Department of Fish and Wildlife, mostly from roadkill and what they call depredation, which is intentional shooting of them when they get and kill livestock or they're a threat to people. So anyway, they have these samples archived. And then the gray ones represent the museum specimens at the UC Berkeley Museum of Vertebrate Zoology, just for comparison. And so we measured the amount of mercury in all of these samples. And fur and whisker, they were normalized to each other to put them on the same scale. Anyway, you can see the dramatic difference here. The ones in blue are about a factor of 3 to 4 higher in mercury content. THG just means total mercury. And when we saw this, we're like, wow, I mean, that's kind of unexpected, right? You have one population that has so much more mercury in it. I mean, granted, that was our hypothesis that they could be picking up mercury from the fog, but I was highly skeptical that this was actually happening. I guess that's a good scientific instinct, right? You want to be skeptical, and I certainly was. But as we dug down into the data, it became more and more clear that there was a food source of this coastal mercury population that was contributing to their mercury content. And we'll get into that in a moment. This is just by age and by region. So the black is the inland ones, and the blue are the coastal ones, and you can see enhancements across every age range. So is this dangerous for the mountain lions? Well, based on the previous slide with the Arctic animals, we would expect observable brain effects to be about at that level. So you can see many individuals would exceeded there. And then a toxicological threshold where it would impair survival or offspring survival would be much higher. However, we did identify 3 individuals that exceeded this level. In fact, the highest mercury content of an individual puma was 22 parts per million, or 22,000 nanograms per gram. And that was in a puma that was found dead, no known cause of death outside of Watsonville. So very, very interesting. More research needs to be done. So we looked into the food web. Of course, puma, what do they eat besides pets? It's very uncommon. Very small portion of their diet is made up of, dogs or cats or even raccoons or feral pigs or goats. 90 to 99% of their diet is deer. Okay, and we have plentiful deer in the Santa Cruz Mountains. Of course, deer are eating plants, and this picture shows deer eating lichen, which I'm going to focus on in a moment. The lichen is an efficient absorber of fog water. Okay, so here comes a puma, and a chase ensues. Of course, the victor gets its spoils and eats the deer. So we were able to get our hands on deer fur samples from the coastal mountains and from inland locations. The deer from the coastal mountains came mostly from the puma kill sites, and from the inland locations from CDFW, mostly hunter check stations. And this is what we saw, very surprisingly. Once again, the coastal deer had about a factor of 2 higher mercury content of their fur. There you can see the mountain lions eating the deer. Okay, so raising some eyebrows now, you can see how the mountain lions might get this enhanced mercury coming from deer. And then we looked at even lower on the food chain, the primary producer, the lichen. Now, of course, we didn't sample all plants, and deer eat many different kinds of plants, but one of their favorite snacks is the lace lichen. It hangs down very low, and it's very nutritious. And it is actually, a lichen is a symbiote between a fungus and an algae. The fungus provides the structure. That's mostly what you see. And the algae is microscopic, but it's the color, the greenish hue, and it actually does the photosynthesis, which feeds the fungus. So it's a symbiotic relationship, and they're very ancient. And Ramalina menziesii is otherwise known as lace lichen, and in 2015, Jerry Brown decreed it the California state lichen. And of course, it's abundant here in the Santa Cruz area. And so we sampled it. And the thought was that is be a very efficient absorber of mercury from the air. Lichen has no roots. It does not collect anything from the ground or from the tree that it lives on. Everything that it gets comes from the air. And it can collect dry deposition of inorganic mercury. There's that Hg2. Or it can collect wet deposition of methylmercury, monomethylmercury, from fog. And it's been used in other studies as a bioindicator of air pollution. So a lot of data on mercury and lichen, but no one's ever looked for fog-borne mercury in lichen. And we thought that this coastal inland gradient might be a good chance to look at that. So this is methylmercury in lichen from 8 sites right along the coast. You can see these coastal sites right here: Año Nuevo, Greyhound Rock, Davenport. I got some lichen off Meter Street near my house. And they all basically fell in the same range. I think the lowest was 16 and the highest was 44. And that's parts per billion. So it's fairly high value for a non-animal. But what was really interesting was that the inland lichen, and you can see with this blue line right here, is kind of the watershed divide, which is also the fog shed divide. You still have fog on the eastern side, but much less and much less precipitating fog. You can see a statistically significant difference there between the ocean-facing sites and the bay-facing sites. So that was also quite unexpected. And what really drove the point home for me was the fact that we saw this same kind of gradient in the deer. So if you look at the deer that came from the bay-facing side of the watershed divide, it had about half the amount of mercury as compared to the deer that were from the ocean side of the divide. So that was pretty consistent with the lichen result. Okay, now the home range of deer, we have to consider this, is pretty small. All right, any one given deer is not going to travel very far, right? So these deer are going to pretty much stay on their side of the watershed divide. Maybe some of these will drop over the other side, but by and large, they're going to stay on their side of the mountain. And so that's why this comparison with this relatively small area didn't work for the puma. You can see there was really no difference between the bay-facing sites where the puma were collected and the ocean-facing sites, because one puma, huge home range, right? Maybe it's not that big, maybe it's that big, but anyway, they overlap. So this wasn't a very good comparison to find the exact effects of fog on puma. But the fact that we see it in lichen and in deer was quite a consistent observation. Okay, and so that led us to this overall diagram here, kind of putting it all together, some of the atmospheric chemistry, the oxidation that we've talked about, the deposition, the getting into the water and the sediments, the methylation, transformation into dimethylmercury, and the upwelling, getting it into the atmosphere and into the fog, where it advects onto the land. You have fog, mercury being taken up to the lichen, the deer eating the lichen, the puma eating the deer. Okay, whereas inland, you don't have that. The mercury drifts inland on the rain, on the atmosphere, and its only deposition is in rain and snow. Okay, you don't have that oceanic influence, and there's no fog. So, and you can see the amount of methylmercury in rain is very low, even coastal rain, because it originates high in the sky and it doesn't go through this ocean influenced fog layer. Okay, whereas mercury and fog is much, much greater. And I guess our hypothesis at this point is that this mercury and fog propagates to the lichen, to the deer, and to the puma. Now, of course, this is a little bit circumstantial at this point still. We don't have absolute proof that this is occurring, but we have enough reason to be concerned about mercury deposition in the coastal environment. And so that led us to our last question here. There's mercury in the fog, so what about our food? Okay, so I'm just going to go through this last section here, and then I'll go ahead and take the final questions. I'm getting near the end, so I want to kind of wrap it up for those people who want to get to the punchline. Okay, so another way in which science works is that you work really, really hard. You do the field work and you do the lab work, and you write a paper and you send it in for peer review, and the editors and the reviewers just tear it to shreds, and you're like, you're hanging your head for like a couple of days, and then you're like, okay, they give you a list of like 4 pages of edits that you have to make on the paper, and it's just a ton of work. But you get through all that and you lift your head up high and you're like, okay, we did it. And then dun dun, they accept it. This little group of like 4 people, they say, okay, this is good enough. That's peer review. And then it gets sent out into the wide world. There can be a press release, or the media can get wind of it, or it can be presented at a conference. You might give a press conference. And so this is what happened. And within a span of about 1 month, I gave approximately, probably an average of like 2 interviews a day, literally probably 50 interviews. Some of them I made links to on my website, which is called FogNet. Somebody sent me a poem called Traces in the Fog. Just some random person. It's a really beautiful poem. Check it out. We had an article in Newsweek, San Francisco Chronicle. I was interviewed for the local TV station. They came in the lab. So fun times. And now in this era of social media, so we had all, traditional media, Newsweek, TV, and, other podcasts and whatever. But then we had just like people talking about it. And some of my students came into the lab one day and they said, Dr. Weiss, your research is just lighting up Reddit. And I'm like, what? No, fortunately, I'd heard of Reddit before, but there was this, a subreddit, UCSC research, and my research was a lot of people were talking about it, and they're like, wow, should we be concerned about the food grown in this foggy area? And of course, it's like the breadbasket of the world here, Salinas Valley, coastal California. And I realized that no one had ever measured mercury content of food from this region. And so I thought, well, I've known some other scientists who have raised money through crowdfunding. And so I thought, what the heck? So tried out a test campaign on experiment.com with the goal of an initial collection and testing of food crops and animal products from our foggy Central California. And we set a goal of $2,000 and raised $2,600 in about 60 days. And you can see there's a list of donors. There's a few family members there, so thank you if you're listening. Appreciate your support. Few friends, but bunch of people that never met before. So what are we going to do with the money? We're working on it right now. Hired an undergraduate, Aaron Gaines, and there we are in the time of COVID. We have reopened the lab, but we have maximum social density in the lab of 2 people and working at any one time. Everyone's wearing masks, but we are getting some good data, and I can share a couple of numbers with you so far. Our first set of results were from some samples that we had taken a while back, and this was of some cow fur from 2 different locations. Went to the Swanton Ranch in Davenport, California, and they put their cows in these little metal squeezers, and I was able to snip off some cow fur, and we measured it for mercury content, and then got some cow fur from the San Joaquin Experimental Range Station outside of Fresno, and we measured the mercury content of that as well. And lo and behold, there's our factor of 2 again. Seems to be a consistent pattern. And these are all grass-fed. They never have any grain, they told me. They're out free-ranging. This is the, the kind of milk and cheese and meat that you want, right? That grass-fed stuff. It's richer and cows are happier and whatnot. Now, I'm not saying that these levels are high, because they're not. Those levels are actually really, really good. Okay, 25 parts per billion, not a problem. So I've shared this data with the ranchers, and they can breathe a sigh of relief, because that, health threshold would be up here, probably at about, okay, that's a number I'd be worried about. But so you can see we're nowhere near that. But it is a little curious, right? The inland cows were lower, statistically significantly so. And now our latest sampling came, also some good news here. We got some cheese at the farmer's market, and this is from a farm in Royal Oaks, California, which is outside Watsonville. And it's from a individual cow named Moonflower. So, and Moonflower is doing really, really well, because in that cheese, there was only even less than the cow fur, 0.4 parts per billion. Okay, so that is extremely low, not a health risk whatsoever. Okay, so good news so far. However, there's much more to be sampled and learned. During this time period, I was contacted by numerous individuals about their concerns about mercury in food. And I show you some of these letters. This person lives in Mendocino, and they raise goats, and the goats eat mostly wild forage, and they do love mosses and lichens, which my family uses extensively for milk and meat. So we're hoping to coordinate with them to get some of their goat sample. And then there's the wild harvesters of nettles, huckleberry, manzanita berries, and so forth. These plants are older, and so they can accumulate more mercury. Part of the reason that the mercury in the cow fur and the cow cheese is probably very low is what the cows are eating. If they're just eating grass, grass is like just an emergent plant, and it just doesn't have much time to absorb mercury from fog or from the air, or really any source at all. So it's not surprising that something that's eating emergent grass is going to have very low mercury levels. And this letter came from Larry Jacobs, Jacobs Farm, Del Cabo. Many of you probably have purchased their brand of cherry tomatoes or their fresh herbs. Or if you've gone to Wilder Ranch, you see some of those herb fields. They grow a lot of rosemary, and they're concerned about mercury in their crops. And rosemary has like a waxy coating on it. And we found plants that have coatings like that can sometimes have elevated levels of mercury. So there again, no one's ever measured it to the best of my knowledge. And so that's something we look forward to doing. Okay, so our overall plan, and this is something that I am excited to share with all of you, especially those of you interested in supporting innovative research. So once we get these first results, that'll tell us what to do next. Hopefully we'll expand testing of crops and animal products grown in the fog belt. We'll increase our regional coverage, Northern California coastal range, north and central coasts. We'll increase the types of crops and products tested. We'll refine some of the variables of importance, like is it a west-facing slope or a certain latitude range? With this data, we'll organize and build a database of results for sharing with the scientific community. We'll build hypotheses and models based on these findings, basically answer the question, which food products are most susceptible? And we will try to see if we can measure and match fog quantity directly with mercury levels. We'll publish the results and communicate with community partners. And of course, this initial work is so important to generate pilot data that can be used to submit proposals to funders National Science Foundation, they funded my original work when I made the discovery of mercury in fog, and US Department of Agriculture, which is important with the health of food. They're interested in the health of food. And of course, we would then begin to understand the impacts of fog worldwide on the cycling of mercury in terrestrial food webs. Of course, there are other fog belts in the world that intersect with food growing regions. So I'm just getting to the end here. I want to acknowledge my co-authors on the study, Mike Bank. He's a big mercury in ecosystems, food webs scientist. Obviously, Chris Wilmers, who does the Puma research. Alicia Torregrossa, who's been very instrumental in fog research, especially in coastal California. And Deanna Clifford with the Department of Fish and Wildlife, who provided a lot of the samples for us. And then my two undergraduates who are now gone, but this work was funded in part by undergraduate awards from the Gunderson family and the Corrett Scholarship. I'd like to thank Carl Lamborg at UCSC for some of his lab support, and individuals out there who have generously donated to the crowdfunding campaign. It's just really encouraging to see so many people really taking science seriously and wanting to have a grassroots approach with it. It's very exciting. And I'll just leave you with some final thoughts here as we watch the fog roll into the San Francisco Bay through the Golden Gate there in time lapse. The discovery of mercury in our fog is a call to action. Much more needs to be done to study this issue. We need to understand the risks to humans in our environment from this process that transfers methylmercury and coastal sea fog to the ground and the environment. And you can help make a difference. Your donation today will allow me to expand my research and help accelerate the understanding of and possible solutions for how mercury in the fog is affecting our coastal food supplies. So help ensure that our local food is safe. And give a little plug there, connect.ucsc.edu. And we have our own little page, Mercury in the Fog, not Mercury in the Foe, Mercury in the Fog. So hopefully you can remember that. And I just really want to appreciate everyone's attention. And if you stick around, I'll do the Q&A now, and then I have a special treat for you. I actually wrote a song titled Mercury. It's my first song called Mercury. People have been asking me to write a song for Mercury for a long time, and so I finally did. And so anyway, stick around. Maybe refill your Stein, and I'm going to refill my water bottle, and we'll have some fun. But first, I'm happy to answer your questions. I'll keep it on screen share in case you want me to go back to a slide. So thank you very much.
Thank you, my goodness. So we have plenty of questions. Keep us busy here for a while, many as we have time for. Tiffany asks, do you know of any research dealing with bioaccumulation of mercury in foraged mushrooms, like oyster mushrooms in coastal California?
Yeah, well, no, I don't. I don't think that's been studied, and that mushrooms are on our list of things to gather. So I believe we will be going out in the first rains. Aaron, the undergraduate researcher, is up for this. I basically say, Aaron, go out in the rain and trample through the poison oak to go get some mushrooms, okay? And he's like, cool.
Okay, Don asks, noticing that cement production can be a significant source of mercury in the atmosphere. Do you have data related to output from local production, cement production, such as the Davenport plant operating for decades?
Yes, we do have some data. We weren't able to find a big signal in mercury in lichen downwind of the Davenport plant. We only collected a couple samples a few years ago, but once again, this is on our list. we want to know really what are all the possible sources of mercury and how they could be affecting plants and food crops in our region. So we are aware of not only the cement plant, there's another cement plant outside Cupertino, which we can sample around. But there's been a history of mercury dumping at the Dimeo Lane landfill from mercury waste from the tannery. We were made aware of this. I'm not sure how well known this is. And for me, it's just hearsay. So nobody say this is a true thing, but somebody told me that for many years, mercury wastes were dumped in that region. So we're gonna go out on a little sleuthing mission and look for the mercury and see how far it is spread.
Next up, we have a question from one of our regulars. Hi, David Cohen. Are the concentrations of mercury in coastal fog in Northern California a function of global mercury emission? Such concentrations begin to as coal combustion declines?
You were breaking up on that. Do you want to try saying it again? I think I got the gist, but why don't you try again?
Oh. Yeah, sorry, Peter. Are the concentrations of mercury in coastal fog in Northern California a function of global mercury emissions, or might such concentrations begin to decline as U.S. coal combustion declines?
Yeah, so I think they are a function of the global emissions, and certainly the U.S. contributes to the global emissions, but most of the mercury emissions in the U.S. are focused east of the Mississippi, and so it really just contributes to that global total as it impacts the western U.S., and the global total is dominated by East Asia. So it's really going to take some adherence to the newly ratified UN Environment Program Minamata Convention, which all countries have signed on to try to decrease the mercury emissions. And of course, the phasing out of coal combustion is really key to this. So some places are having more success with that than others.
Thank you. Next question from Julie. I remember an ecologist hypothesizing that mountain lions were eating a number of wild boar, especially in years that the deer population dropped. Any thoughts about the boar connection?
So once again, you're dropping out a little bit. The wild boar, the mountain lions do predate on them, but it doesn't make up a consistent part of their diet. I'm not sure if they would have higher mercury than the deer, but it would be really cool to get our hands on some boar fur so we could establish that.
Okay, I'd like to move on to a question from Jamie. Since mercury affects brain function, have you come across any correlation between mercury levels and behavior, such as wandering away from their typical habitats into neighborhoods or places they don't normally travel to? I assume they're talking about mountain lions rather than the people.
Well, one would expect that. I don't think there's any direct proof that that is occurring. I know there's been several mountain lions that have wandered, into urban areas, and they may have elevated mercury levels, but there's also many other pressures, habitat fragmentation and loss of genetic diversity are the two primary risks to mountain lions. So, mercury might be making them a little bit stupid and having bad judgment. For some with the very highest levels, their offspring are probably not going to survive. But yeah, there's just too many unknowns to really draw a direct cause and effect.
Okay, we have one from Daniel. In northern Chile, a major source of supply of water comes from harvesting the fog. They also harvest water from the fog to irrigate the grapes that they use to make pisco. Given that this is a similar upwelling system, wouldn't you expect elevated mercury in people near in the grapes? And we have somebody who wants to go to Chile to study precisely this. She volunteered, or excuse me, and they also wonder about grapes and Coralitos then.
Yeah, well, we're going to test grapes. And grape plants are older. You know, if you think about the age of a plant, that gives it more time to absorb mercury. So that emergent grass is going to be really clean. But, you know, grape leaves are probably higher. The grapes themselves, I mean, I don't know. They're mostly water. So that is going to be a benefit for lower mercury. I would expect wine and grape juice and grapes to have relatively low mercury just because of the high water content. But would there be more mercury in foggy wine than in non-foggy wine? Yeah, quite possibly. And could that show up in humans? I mean, maybe. Humans eat a lot of different things. So, you know, animals are going to be the biggest source of mercury just because of the bioaccumulation factor, and plant matter and fruit matter is going to be a lesser source, is what I think.
I'll try one more time. I hope you can hear me, Peter.
Okay.
Question from Tiffany. Is there any research on the mercury concentrations in other high-level coastal terrestrial predators like grizzlies or wolves further north in BC and Alaska?
Yes, there is. There's been work on grizzly bears, but wolves, I don't think so. And other terrestrial mammals, no. The grizzly bears have an aquatic diet. So they have elevated levels of mercury just simply because they eat a lot of fish. And a lot of the fish that they do eat are salmon, which go to the ocean and can collect more mercury that way. So right now, we don't have a direct analog to the Santa Cruz mountain lions of a top predator from a purely terrestrial system. But that would be an area of future research to maybe get some samples from wolves, coyotes, you know, lynx, that kind of thing.
Thank you. And a question from Maura. Can animals or humans also acquire mercury via methods other than eating contaminated food, such as breathing in fog? For some toxins, at least, aspiration seems to be an effective mode of acquisition in the body, e.g. chloroform and chlorinated shower water can make a person ill or even unconscious.
Mm-hmm. Yeah. I think the concentrations are just too low in the atmosphere for that to be any kind of a health risk. For mercury anyway, but for other substances, you know, much more. I mean, they're bubbling like pretty high concentration chlorine gas through municipal water. So it's going to make higher concentration, you know, chlorinated byproducts than what we have of mercury compounds in the air, which are extremely dilute.
So Molly wonders about redwoods. Has anyone studied the effects of mercury in fog on redwoods?
We've measured mercury in redwood needles. It is higher than other plants, much higher. Bay leaves are actually even higher than mercury needles, it turns out. I actually wouldn't eat bay leaves. They were kind of high, getting upwards of the higher than we saw right about the same as lichen, actually. So, you know, kind of up there. As far as toxicity to the plant, I'm not aware that mercury causes toxicity in plants because they don't have the same kind of proteins that animals do. They don't have the neural function, right? I mean, plants have feelings, I suppose, but their neural networks, their neural cells are very different. So.
Laura wonders, to what extent are the chemicals used in conventional agriculture contributing to the mercury that ends up in the ocean and subsequently in the fog?
To my knowledge, mercury is not being used in the fields. So I don't think that's a very big deal, and runoff in general is not that important because the mercury that ends up in the water and the sediment, it tends to be fairly fixed in place. It doesn't mobilize and enter the fog that readily.
We have about 5 more minutes, so it looks like we have all kinds of questions pouring in. I apologize, we won't get to all of them, but we'll try the best that we can. If you want to upvote questions that you think are especially important, go ahead and do that. I'm going to take a question from Michael Beasley, which just disappeared. Have you considered collaborating with the UCSC agroecology program to study how mercury and fog might impact farms?
Did you say the agroecology program?
Yes.
Yeah, we are in the process of connecting with that program to obtain some of our crop samples. And so hopefully our discussions will lead to a further study and help us to have access to other samples from other farms and to find any differences that, explanations for differences that we discover.
Yes, thank you. And I see several questions in here about recording the talk. The answer is yes, the talk is being recorded. In a couple of days, it will appear on the UCSC Arts and Lecture page, and we will be sending a link out to that in our social media and thank you posts. I think there's one question here, if other people can use this. Peter, you want to address that? This is all public research. And present your YouTube page.
Sorry, you were breaking up again. Is it about my FogNet website? Is that what you're?
Question is, can people? So this will be posted on the YouTube channel. I assume this is all public research.
Yeah.
Will use this lecture in classes or otherwise?
Yeah, I do. Yeah, when I introduce my students to my work, I show a lot of these same slides.
And we have a question from Bill. I'm sure that you are aware of the high mercury levels in elephant seals. Have you done your research to those efforts?
Yeah, so the elephant seals are eating fish, and those fish are in the ocean, and that's where the mercury methylation is occurring. So that is kind of a separate zone, you know, just staying in the ocean right now. And those elephant seals go on to land, they molt, and their fur contains very high levels of mercury, and it can actually contaminate that coastal zone. But our research is looking at kind of mercury jumping out of the ocean, going into the atmosphere, into fog, and then coming into land and falling down in a purely terrestrial environment. So connecting all of the different mercury research is a challenge, and making sure it, you know, all kind of fits together, because it's the same element and it moves around and changes forms, and there's always a surprise with mercury.
So one of the surprises has to do with impacts on brains. How does mercury affect brain cells? Does it lead to Alzheimer's, Parkinson's? This question's from Sylvia.
Yeah. You know, I wish I had a really clear answer on that. First of all, it's not my specific area, so I don't want to start quoting things that I'm not an expert on. But I don't think that there is a link between there's not a proven link between Parkinson's and Alzheimer's and mercury. Now, there's maybe some evidence suggests a correlation, But the only thing I'm aware of as far as the neurochemical problems or the pathology is just in motor coordination, speech impediments. It can lead to lower IQ, other direct problems with that they've observed in people who've been, chronically or acutely exposed to mercury.
Okay, and our final question for the evening, I'm sorry to say, Raphael asks, sounds like you have data on the travel range of individual pumas. Can you limit your analysis to comparing pumas who only spend time on one side or the other side of the mountain?
Yeah, no, that's a good idea. I'm not sure that our sample size would have been large enough. Yeah, good. That's a good point. We can dig down in the data and see if something emerges.
Well, thank you all for coming tonight, and thank you, Peter, for joining us for this evening. It's been
Yeah, my
wonderful, I
pleasure.
have to say. Thank you. As Mike mentioned, it's been recorded. It will be up on our channel. Oh, I'm sorry. Yes. Oh,
I don't want
go for it.
But here we go. I don't need my doodles. And here we go. This song's called Mercury. And it goes like this. Known about it since antiquity, a shiny metal that is liquidy. Its symbol stands for hydrogyrum, Latin word for water and silver, then combine them, mercury. Its claim to fame is the amalgam. It forms with flecks of gold when it's around them. This gave birth to hydraulic mining and the need for more mercury refining. Mercury, mercury makes you go berserkery. Mercury makes you go berserkery. Super fun sights so bad they make you shiver. San Francisco Bay and Carson River. The microbes perform the methylation. Then look out for bioaccumulation. Mercury. For mercury, dilution is no solution. The smallest amount can cause serious pollution. We'll find some safer techniques for mining. Coal-fired power plants will get some serious redesigning. Mercury, mercury makes you go berserkery. Mercury makes you go berserkery. Yeah. Yeah, it's mercury. Oh, mercury.
Marvelous. Thank you. So before we leave you, Peter, again, thank you for the talk, but do you have any ask for our participants tonight?
Well, yeah. Right now we're just working on a shoestring, and if you believe in this, the importance of this research, and want to support it. You know, we could use in the neighborhood of tens of thousands of dollars, up to 100,000 spent over 6 months to a year. It would allow me to hire full-time research scientists and support more graduate students, and that would help us expand our efforts. It would also make us more likely to attract funding from government agencies, because we'd have a more convincing data set, better interpreted, analyzed, and considering more of the questions that you all have raised here tonight, which really drives the science forward. It's amazing the different perspectives that we can get from just talking about it with the general public. So I really appreciate that. And yeah, right now we're operating on very low funds and getting the most out of it, but we're certainly looking to expand the testing into some of these things that people have brought up, the different types of samples, the different sources, whether it's cement or mining, to really establish what are the risks to people and the environment when it comes to mercury. So great, thank you for asking, and I hope that provided an answer for you.
Well, thank you again. That's marvelous. Very much enjoyed it. All right, we have one more piece of art to share tonight.
Mm-hmm.
This is our newly launched Slugs and Steins logo. So there, this was produced by UC graphic designer Miranda Powell. Thank you, Miranda. One more thing, I guess you can add into the Q&A your comments for Peter if you want to send any further questions to him that way, but also appreciation for the talk. Okay, big round of thanks also to the staff of the alumni and special events offices who set this up. Thank you, Kara Haley, Diana, Paulina, Kristen. Schedule going forward. Our next event, Monday, August 10th. August 10th, same time, same place. Will feature Associate Professor of History, Greg O'Malley. Greg is specialized on the history of the slave trade. He earned his PhD from Johns Hopkins, joined UC Santa Cruz faculty in 2008. His 2014 book, Final Passages, The Intercolonial Slave Trade of British America, 1619 to 1807, received 3 book awards and has smoothed the path for multiple grants. Recently received funding to expand coverage of the intra-American slave trade database to encompass the forced movement of enslaved people born in America. So join us for that. Again, Monday, August 10th. In between, all summer long, if you're into Shakespeare, we have Undiscovered Shakespeare, War of the Roses. This will be on every Wednesday this summer, 6:30 to 8. The University Forum will have yet another, we'll have more on the COVID. So Lessons of COVID for Global and Community Health. That'll be next Monday at 5:30. Then we'll have a CRA lecture on UCSC's COVID-19 Diagnostic Lab. That'll be on the 23rd at 5:30. So again, thank you so much for joining us, and we certainly hope to see you back on August 10th for the next virtual event.